We are advancing treatments for glioblastoma multiforme, a rare but extremely aggressive and deadly tumor of the central nervous system. My name is Adam Robertson, and I am the Chief Scientific Officer at Hemispherian. Let's begin by discussing why glioblastoma multiforme is so bad and is among the highest disorders with the highest unmet needs in oncology. 5-year survival is extremely rare, only about 1% long-term survivors. Median survival with treatment, radiotherapy, and with surgical resection is about 12.1 months. This is only improved by about 2.5 months by the most aggressive type of chemotherapy. And the reason for this is the frontline therapeutic. Most patients are resistant. 3/4 of all patients receive no benefit from the frontline therapeutic. So therefore, only 1/4 of patients have any treatment available to them at all. And this is why this is among the disorders with the highest unmet needs in oncology. So let's talk about the compounds that we're developing. Our compounds target a new mechanism. They are well tolerated, and they cross the blood-brain barrier very readily. This is based on about 10 years of academic research in my academic laboratory. My academic laboratory was interested in a particular DNA-based modification called hydroxymethylcytosine. And we noticed that hydroxymethylcytosine was very strongly correlated with cell doubling time. If you look over here on the left, brain cells have a high amount of hydroxymethylcytosine and they have a long doubling time. Whereas if we drop down a little bit further, testes and spleen, which have cells that divide very rapidly, have low amounts of hydroxymethylcytosine. Further, we see over here on the right that as cells become precancerous and then cancerous, they have decreasing amounts of hydroxymethylcytosine. This is even among cancerous cells that you can see that their prognosis is based on the amount of hydroxymethylcytosine. It can be determined based on the amount of hydroxymethylcytosine present. Poor prognoses have low amounts of hydroxymethylcytosine. My laboratory ended up publishing a few papers on this, and we actually have a few patents that identify hydroxymethylcytosine levels. In addition, this data is corroborated by clinical trials, which essentially show the same thing, that cancerous cells have low amounts of hydroxymethylcytosine, and you can determine cell types that have cancers that have poor or good prognosis by the amount of hydroxymethylcytosine. But what we're interested in here today is actually treating the tumor. So what we thought initially was that if we were to take a cancerous cell and increase the amount of hydroxymethylcytosine, we should be able to slow its doubling time. So to test this, we decided to overexpress TET2. TET2 is the enzyme that makes hydroxymethylcytosine. So over here on the left, we see that we increase TET2, and then if we increase TET2, we see about a doubling of the amount of hydroxymethylcytosine. And when we see that doubling of hydroxymethylcytosine, we see, as expected, we see a cell cycle delay. The cell cycle is delayed by about 3 hours, and this is also most pronounced in a G1 delay or G1 arrest. So we can target that with genetics, so that's neat. So this is something we can try to then target with small molecules. And here's what we're looking at. Cancer cells have a low amount of hydroxymethylcytosine. So if we target these with small molecules, we should be able to increase the amount of hydroxymethylcytosine. And after increasing the amount of hydroxymethylcytosine, we get a cell cycle delay. And then based on other data, we see that these cells should die. Normal cells, however, have a relatively high amount of hydroxymethylcytosine. They're not would be targetable by the same type of drug because all of the substrate would be blocked. This would not result in a cell cycle delay. They would actually behave normally, and things would be fine. So we have a great situation here. If we can target this particular mechanism with small molecules, we can target cancers, but not normal cells. So let's take a look at the first data that we saw. We ended up searching through many molecules and to test this, and we found 2 molecules that were very good, that were targeting this mechanism and also killed cancers. We call these GLIX1 and GLIX5. If we look over here on the left and in red, so red is cells that are dead and varying degrees of dead, and green is cell types that survive. GLIX5 kills a broad range of cancers, as would be expected, and GLIX1 kills a narrower range of cancers. Most notably, the cell type that's killed most readily and best efficacy is gliomas or glioblastomas. So what we did then is, because we see our best efficacy in glioblastomas, is we took a bunch of patient-derived glioblastoma cell lines and we tested GLIX1 and GLIX5. And we saw that we killed about half of the glioblastoma cell lines that we tested. And if we paired this with temozolomide, we could kill roughly 75% of all glioblastoma cell lines. Now, importantly, and a good drug kills the cancer, but doesn't kill normal cells. So that was the next thing we tested. We tested normal cells, and we saw that, in general, GLIX1 and GLIX5 do not have a lot of cytotoxicity towards normal cells. So we would not expect to have a lot of side effects. GLIX1, being our lead candidate, has almost no cytotoxicity to normal cells. So that's great. We can see this effect in cell lines, but we wanted to jump into xenografts, a better model for testing anti-cancer agents. So in this case, we took 24 mice and 24 tumors. 24 tumors were implanted, and these were allowed to grow to a predetermined size. Once the tumors reached that predetermined size, they were randomized into 3 different groups. The control group, where we saw that the tumors grew relatively large and caused negative effects for the mouse. If we treated with GLIX1, we saw regression in 8 out of 8 tumors. 4 out of 8 were regressed to the point that no tumor was detectable at all. And if we treated with GLIX5, which targets this mechanism also, we see regression in 8 out of 8 tumors. And this is quantified over here on the right. We see that we get significant tumor regression by targeting this mechanism. We also had this validated externally. GLIX1, we sent GLIX1 and GLIX5 to neurosurgery at the United States National Institutes of Health. We spoke with the chief of neurosurgery there. He really liked the data that we showed him, and he decided he wanted to test this in his model. So this is a very patient-derived neurospheres, which are essentially the closest to being able to treat a human brain tumor, aside from actually treating a human brain tumor in a patient. And we saw that we're able to kill all 3 of these cell lines that we tested. So we can validate this works at the National Institutes of Health, at the CRO that we asked to do the xenografts, and in our own laboratory. We also show that we get limited side effects, even at very, very high doses. So if we repeat dose mice with up to 2,000 milligrams per kilogram of either GLIX1 or GLIX5, we see that these mice continue to gain weight and they behave normally. There's no overt side effects, and everything with these mice appears to be normal. So by targeting this mechanism cancer, we can kill the cancer, but have very limited or no side effects in the mouse. And this would be expected to be true for humans as well. Another important feature is that these drugs do pass the blood-brain barrier. GLIX1 and GLIX5 are very efficient at getting across the blood-brain barrier. At the same time, antibodies, this represents a significant advantage over antibody-based therapies, which do not pass the blood-brain barrier. So we do not have a significant amount of competition from antibody-based therapies for this particular indication. We own 2 patents for this. One is for the compounds, and the other is for a synthesis method of the compounds. I'd be happy to show you these patents under a CDA. So this is our plan, our progress plan. This is, as I mentioned before, based on about 10 years of academic research. This cost about $10 million to fund over the past 10 years. And since Hemispherian has acquired the rights to this, we have spent about $250,000 of our own money to advance this project. Right now, we're trying to raise $12.5 million in 2 tranches to complete an IND enabling package and a phase 1 trial. At the end of the phase 1 trial, we expect to be keenly positioned for an exit or to raise more capital for a phase 2 trial. The 2 tranches, with tranche 1 is $4.7 million that we're trying to raise, and this will help us to complete an IND enabling package over the next 12 to 18 months. And we anticipate that a phase 1 trial will cost $7.8 million. This will be tranche 2, and that will it will be completed after 3 years from today. And as I mentioned, we'll be in a great place for an exit or to continue the project. So what we're looking for right now is about $12.5 million to complete an IND enabling package and a phase 1 trial. Both of the inventors are on the team, and we've partnered with Hemispherian founders from Hemispherian and other Hemispherian scientists to make this project a reality. Here's my contact information. Please feel free to drop me a line or give me a call if you're interested in discussing this project. I would certainly be happy to have a follow-up phone call or a video conference to discuss this. Thank you very much, and I look forward to hearing from you.